Good. Well, I'm glad everybody's back from their break. So the last talk of this is on the COVID vaccine. And I think IAS-USA has always been very prescient in terms of starting with HIV, moving quickly to HCV, and now COVID, among other viral diseases. And we're pleased to have Ann Falsey with us. Ann really comes from a dynasty in Rochester, which is focused on viral diseases for, I don't know, Ann, probably about 50 years or maybe longer, starting with RSV and influenza, now expanding outwards. And Ann has done a lot of work on RSV and metapneumovirus and now COVID. And I think that if there's one topic in virology everybody wants to hear today, it would be what's going on with the vaccine. So, Ann, you have to tell us everything in your allotted time.
Well, thank you, and thank you for inviting me to this meeting. I like to tell people it's either a really good time to be a respiratory virologist or a really bad time. So I'm going to give you a whirlwind tour of what's going on with the COVID-19 vaccines. These are my disclosures. And, I'm pretty sure everybody listening to this talk is aware about how bad this epidemic is, but I like to show this slide in that we who are in respiratory viral infections look at these sine waves, as you can see. It's called the PNI graph, and so every winter season there is seasonal variation with an increase in respiratory and infections and pneumonia, and that when the excess mortality exceeds that expected baseline, we call it an influenza epidemic. And you can see in 2018 here, we did have a pretty bad flu year, but if you look over to 2020, it's absolutely dwarfed by COVID. So they've now included COVID and the deaths due to COVID. It's just monstrous compared to flu. So we all know it's a huge problem. So before talking about a vaccine, we really need to just review a little bit about the virus. And so many people know that it is a coronavirus. It's an RNA virus. There's a lot of different animal species throughout the animal kingdom, and particularly bats. Bats carry everything that's bad, and other animals are then intermediaries. It got its name from this the surface, which reminded somebody of the spokes on a king's crown. These are the surface proteins, the spike protein, and they thought it looked like a corona, a crown. There are 7 known human strains, and listed here are the 4 ones that really cause seasonal disease. So bronchitis, pneumonia, colds, and things like that. And then at the bottom are the 3 that have emerged more recently, causing severe respiratory distress. So as I mentioned, all bad things come from bats, and the current coronavirus has 89% gene identity with a bat-like virus. And this is the phylogenetic tree. There are alpha coronaviruses and beta coronaviruses. And over here are the bad ones, the bad actors, and this is SARS-CoV-2, which actually is phylogenetically fairly close to SARS-CoV-1. This is a bit of a cartoon which allows us to review the structure. It is an enveloped virus, and here is this all-important spike protein. And this seems to be the major target of the immune system and neutralizing antibody. And so all of the major vaccines that are moving forward are directed against making antibody against the spike protein. So a little more detail about this spike protein. It's a class 1 fusion protein, just like RSV. And on the top here that's shown in green is the part of the spike protein that actually binds with the receptor binding domain. It's referred to as the receptor binding domain because it binds to the ACE receptor, which is on the respiratory epithelial cells. And this little green part of the protein can extend and sort of stick out its thumb when it's going to bind. And so this, again, is just a cartoon. This is the prefusion spike. And one thing to say about fusion proteins, they do exist in a metastable state when they're pre-fusion, before they have fused with the cell. It sticks up its thumb, it binds to the ACE receptor, and that's when fusion is initiated, and there is some fairly significant change, structural change, in the protein after it fuses. And with other viruses such as RSV, the pre-fusion state seems to induce more robust neutralizing antibodies. It doesn't seem quite as important with the coronavirus spike protein. But you could see if you had an antibody that bound to this portion of the spike protein, it would really interrupt binding and biological activity. So, how do we get out of this mess? Herd immunity and vaccination are really the only two strategies. And obviously, herd immunity is not acceptable. It just accepts that, millions of people are going to die. So that's why we're talking about vaccination. But again, before you get to vaccination, a lot of coverage in the news particularly is, well, do we really have immunity to SARS-CoV-2? Is it possible? And you will hear things about people being reinfected after actually having coronavirus. A lot of that is simply the tail end of a prolonged low-level excretion, and it's not really reinfection. But there have been some documented reinfections when they've done sequencing of the virus. Interestingly, these are people that are basically asymptomatic and were picked up on screening. So I actually have no reason to think that this coronavirus would act markedly different than the other seasonal coronaviruses, where partial protection is a good thing. All of the respiratory viruses don't induce lifelong sterilizing immunity. If you think about flu and rhinovirus and RSV, we get them when we're little and we get an infection, and then we have a period of partial immunity, and then as the antibody wanes and immunity wanes, we can get sick again, with a cough or a cold or bronchitis, and generally you don't end up with a severe pneumonia and on a ventilator. So I think that would be the most likely scenario, but since we don't know, we don't have experience. There's amazingly little that is written on the immunity to coronaviruses, and I think it's because nobody cared when they just caused colds. And this is one study where they looked at a challenge model with coronavirus 229E, and they took 33 people, and they drew blood at baseline, and then they actually challenged them with the coronavirus. And they looked at neutralizing antibody, serum binding antibody, and the nasal antibody, and they compared their volunteers that had mild illness to those who had moderate illness. And notably, those who had moderate illness, more severely ill, had significantly lower pre-infection antibodies than did the people with mild illness, suggesting that high antibody levels are beneficial and can ameliorate the severity of disease. So another concern when talking about vaccine development is mutation of the virus. And this is a graph where they pictorial look at that with each change in color, it represents a mutation. And RNA viruses are notoriously mutatable. And so this is back in December, and you can see that as it changed color over the ensuing months, that there are a fair amount of mutations. Most of these are silent mutations. But one thing that's interesting is the aspartate to glycine mutation at site 614. So the original virus in Wuhan, China, is shown here in green. And then as it moved over to Europe, there was a mutation at this site. And then this has actually become the dominant virus throughout the world. And there's just been very recent data in animal models that show that this mutated virus actually replicates better in the upper airways. So no surprise that it would become the more dominant strain because it transmits better. But interestingly, the antibody that is generated from many of the vaccine candidates, it neutralizes both strains equally well. Lastly, the thing we worry about is antibody-enhanced disease. And This really came to light with the RSV vaccine in the 1960s, in which they used sort of a brute force. They just formalin inactivated the virus. And unfortunately, what this did was produce a fair amount of non-neutralizing antibody. And there was also a shift from a nice balanced immune response, humoral and cellular immune response, to a dysregulated inflammatory response. So if you look at the cartoon, neutralizing antibody actually prevents the virus from binding to the ACE receptor, whereas non-neutralizing antibody does not neutralize the virus, and the FC portion can then bind to FC receptors on monocytes and macrophages and actually facilitate virus entry into the cell. And so for the coronaviruses, this is a theoretical concern. There doesn't seem to be any evidence. All of the vaccine candidates moving forward have had scrutiny for that they're inducing a good ratio of neutralizing to non-neutralizing antibody, that they're have a Th1 biased immune profile, and there is a requirement for animal models to show that with challenges there's no immunopathology that's observed. So I think we all know that in normal times, vaccine development takes a long time, 10 years, to go through the preclinical stages and through phase 1, 2, and 3. This has been like nothing I have ever experienced, but it's important to note that the stages are not actually being skipped. Each stage is being done. It's just being done very rapidly with everybody working 24 hours, 7. There's no such things as weekends anymore. And the regulatory bodies are turning things around very quickly. So, in steps Operation Warp Speed, which has the dumbest name ever, but it's actually a good thing in that not only is it funding some of these very big phase 3 trials, it's allowing the vaccine manufacturers to produce at risk. So they are gearing up and producing large amounts of vaccine without actually knowing that it's going to be effective, because the last thing you want to do is reach a point where you show the vaccine is great, it works, and then you have to wait 6 months for them to gear up manufacturing. The 3 main types of vaccines that are currently in phase 1, 2, and 3 trials are listed here, and they're all different ways of skinning a cat. They're all aimed at making antibody to the spike protein. So there are the messenger RNA vaccines, and this is where basically the mRNA, the naked gene for the spike protein, is encapsulated in lipid, and it provides the instructions once it enters the cell for the body's cells to make the spike protein itself. And this approach allows the body to make the protein, so we don't worry quite as much that we may be disrupting important epitopes and making non-neutralizing antibody. It also is apparently easier to scale up large volumes of vaccine. The second approach delivers the spike gene protein using another organism, typically an avirulent, non-replicating virus. It's sort of like the Trojan horse approach, and very frequently adenoviruses have been used for this type of vaccine. And then lastly is the spike protein is just made in the lab, and that's kind of the traditional approach. That's how flu vaccines are made. And I'll just briefly go through some of these different vaccines. These are the vaccines that are in development listed here. Moderna and Pfizer are RNA, Astrazeneca and Janssen are vectored vaccines, and Novavax and Sanofi are purified proteins. Interestingly, Pfizer got impatient and just opted out of Operation Warp Speed. I think basically because they didn't want the government telling them what to do, and they wanted to just get going and not wait in line. So all of the studies going forward through Operation Warp Speed have been relatively harmonized. They are large sample sizes, 30 to 60,000, to allow a fairly quick result. They are event-driven, so that means that you need to achieve a certain number of cases to look at how many cases occur in the vaccine recipients versus the placebos. And very typically, there is an interim analysis built in at the halfway point. The primary endpoint, they differ a little bit by trial, but it's really symptomatic PCR-proven COVID disease. And, the estimates are a little different with each study, but they assume at least a 70% efficacy and generally assume somewhere between a 0.6 to 1% attack rate in the population. And estimate, given that, you would need about 6 months to accrue cases for your final analysis. And obviously, that could change depending on, the way things are going, at least in the U.S., we may actually get to our answers sooner because the attack rates are starting to be very high. And notably, there is a single data safety monitoring board that is monitoring all of these studies, again, to look for any trends, particularly events that may be low numbers in each study, such as enhanced disease. They can look across the studies to look for any safety signals. So the first one out of the gate was Moderna with a messenger RNA vaccine, and they published this in July. And this is just an example, and I won't show every single study, but this is their phase 1 reactogenicity study. And you can see here that severe is shown in yellow, blue is moderate, and gray is mild. And these are the systemic symptoms, and down here are the local symptoms. This is vaccination 1, vaccination 2. And, just the eyeball test shows you that, they're getting more reactogenicity with the second vaccination. But if we also look at the different doses they were using, 25, 100, and 250, that most of the reactogenicity, the severe, is in that 250 microgram dose. So when they looked at their immunogenicity, these are the 25, 100, and 250 microgram doses. And this is after the first vaccination, and this is after the second vaccination. And you can see there is an immune response with one dose, but you really boost nicely with a second dose. And you can look over here, and this is 100 micrograms, and this is 250 micrograms. And most of the studies provide convalescent sera as sort of a yardstick to measure these, that there really is no difference between the 100 and the 250. And so because the 250 was really unacceptable in reactogenicity, they moved forward with the phase 3 with the 100 microgram dose. The Pfizer constructs, they looked at the just gene segment with the receptor binding protein, and then they looked at the whole spike protein. This does contain mutations to hold it in the pre-fusion state, and then they are coated in lipid, and that helps to get it into the cell better. And in Rochester, we were fortunate to participate in the phase 1 study. There were 4 U.S. sites, NYU, Maryland, Rochester, and Cincinnati. And there were 150 people enrolled, and we evaluated 2 different constructs that I just showed you and 3 different doses. And this data was recently published in the New England Journal of Medicine. And the reactogenicity, interestingly, was less with the whole gene compared to just the receptor binding domain. And then this shows you the neutralizing antibody responses. These shown in yellow is the B1 construct, that's just the receptor binding, versus purple shows you the whole gene. These are the young folks, these are the older people. And again, the neutralizing responses with the 30 microgram dose were, essentially just as good as the other construct. And because it was less reactogenic, that was the dose and construct that was chosen for the phase 3 trials. So both the Moderna and Pfizer trials began on July 28th. Moderna had 99 sites, Pfizer 149 sites with some international sites. These are both 30,000 per, well, Pfizer actually increased its enrollment to 45,000. But they are 1-to-1 vaccine to placebo, and the endpoints, again, are symptomatic COVID-positive illnesses, and they estimate a 6-month time to final analysis. So moving on to the vectored vaccines, these 4 shown on top are those that are moving into phase 3 trials. the Chadox, AstraZeneca, the Johnson. And then there is a Chinese vaccine made by CanSino, which is adenovirus 5. And then there's a heterologous prime boost with an adeno 26 and Ad5 made by the Gamela Institute in Russia. These are just a list of some of the preclinical vaccines that are being moved forward that are vectored. And there's a number of organisms that they're using. And I just call your attention to deactivated rabies. And I don't know whether that's a good idea or not, but I think it would be a nightmare for the marketing people. So moving on to AstraZeneca, this is the otherwise known as the Oxford vaccine. Their phase 1 data was presented in Lancet. This is a replication-deficient chimpanzee adenovirus, which expresses the spike protein. It is not stabilized in the prefusion form. And a chimpanzee adenovirus is not pathogenic to people, and hence they are not immune. There's a fair amount of experience with this vector. It's been used with flu, TB, malaria, Zika, and most recently MERS. And so the phase 1 data was in roughly 1,000 people. They had a control vaccine of Menactra. It wasn't a saline placebo. And originally they intended it to be a one-dose vaccine, so they only did 10 people with a prime boost. And this is the dose, 5 times 10 to the 10th particles. And this is just the reactogenicity data. On top is first dose, second dose, and this is local, first dose, second dose. This is systemic, with yellow being mild and this pink color being moderate. And with the vector vaccines, the reactogenicity tends to be more intense with the first vaccination than the second, unlike the RNA vaccines. And it was basically felt to be quite acceptable in terms of a reactogenicity profile. Again, this is just an example of their immunogenicity data. This is at day 28 after one dose, and they did find, and this is only 10 people, that they did get a significant boost if they gave another dose, and this here is convalescent plasma. So in the end, they stopped hurrying, and they went and did prime boost in all the different age groups, and it looked significantly better, so they went with that for the phase 3 trial. So the phase 3 trial of AstraZeneca is 2 doses, 28 days apart. In this study, 2/3 of the participants get the vaccine versus the placebo. And again, the primary endpoint is symptomatic PCR-positive disease. The U.S. study opened on August 31st. 829 people were enrolled. It was paused for 7 weeks because of a neurological event that occurred in the UK. It was thoroughly investigated by the British authorities and the FDA and was reopened to enrollment on October 28th. And so far, probably today, there's about 6,000 people enrolled. Moving on to Janssen, this is an adenovirus 26 vectored vaccine. It includes the spike gene to code for the prefusion spike. Because adenovirus 26, which is a human pathogen, it's an uncommon pathogen, though, and so there's not a lot of pre-existing immunity in the human population. Indeed, there is also experience with this vaccine for other pathogens, Ebola, Zika, HIV, and RSV. So this is their phase 1 data. It's just in preprint form, as far as I know. They did 2 cohorts. They did young and older, and then they did 2 doses, a low dose and a higher dose. And they also are doing 1 dose and 2 doses, but it's 56 days apart. And so their data that they present is really just 1 dose. And because I think that it's a balance between getting going and getting people protected versus optimal immunogenicity, they decided to go with a 1-dose vaccine. And this is just the data from that study. Cohort 1A is young, cohort 3 is the older people. Here are the 2 different doses, 5 times 10 to the 10th, 1 times 10 to the 11th. And it shows their antibody responses to a single vaccination. Again, convalescent plasma is your yardstick. And, they get a pretty good immune response. But if you look over at convalescent plasma, with the one dose, it does tend to be on the lower side of what you can achieve with natural infection. So they've begun their phase 3 trial. It opened September 23rd. It's a very large global study, 60,000 people. They ended up going with the lower dose because it didn't seem to have a significant effect on immunogenicity. And this is placebo vaccine 1-to-1. The Chinese vaccine, the Ad5, is going forward, and this is just an example of their phase 2 data. They did look at pre-existing antibody in the population to Ad5 because it's a fairly common serotype and people have immunity. And they did notice a significant negative impact with increasing age and antibody greater than 200 to Ad5 in the population. There's also been concerns because of the experience with the HIV vaccines, where there seemed to be an increased HIV infection rate in those people with pre-existing immunity with an Ad5 vectored vaccine. So, in many groups, there's a concern using adenovirus 5. And then lastly, I just mentioned the Gamela Institute vaccine. This is the Russian vaccine. Oh, and I should say the Chinese vaccine is in phase 3 in Pakistan. So, you may have heard on the news that, Vladimir Putin approved for use under emergency use their vaccine. It's interesting. I'm not going to bother to present the data, but their phase 1 data was published in Lancet. It's only 76 individuals, and they use a priming with the spike protein with an Ad26, and then they boost with Ad5. The immunogenicity looks fine, but they basically approved it for use before they've done their phase 3 study, and that is reportedly ongoing. So lastly, the protein-based vaccines, this is, the real traditional approach. You need to be very careful to preserve the natural shape of the protein so that you don't somehow interrupt or inactivate the important epitopes. Frequently, adjuvants are needed to just provide that extra boost to the immune system. And this is the technology used by Sanofi Pasteur. It's called the BEST platform. And what they do is they engineer in the spike protein to a baculovirus. The baculovirus then infects insect cells, and they can do this in bioreactors, and then they purify the actual protein, and that is for the vaccine. The two companies using this approach are Sanofi Pasteur. Interestingly, they've partnered with GlaxoSmithKline and to make the adjuvant IAS-USA, which is used in Flublok. It's anticipated the trial might start in January 2021. Depending on what is approved at that point, it may have to be an equivalency trial. And then Novavax is also using spike protein in a nanoparticle with an adjuvant called Matrix M, which is a saponin-based adjuvant. They have had some success with their Ebola vaccine, and this trial is scheduled to begin sometime in December. So it's a real horse race, and, initially Moderna got out of the gate maybe a little faster. Pfizer has been working hard to catch up, so I'm sure that you all heard the news about the Pfizer interim analysis that was just a few days ago. So they've had 43,000 people, a little over 43,000 people enrolled, and so at their interim analysis of 94 cases of COVID that occurred at least 1 week post the second vaccination, it was found to be 90% effective to prevent symptomatic infection, which is really quite extraordinary. Interestingly, the Data Safety Monitoring Board recommended that they continue the study to the final analysis of 164 cases. And you might ask why. There's so few cases in the group that received the vaccine, so sort of vaccine failures. I think that it's important to make sure that there's no hint of enhanced disease if you're not completely protected. And we have to remember we're going to be putting this into millions of people, and so it's important to do all the proper safety testing. But we do know that they plan to file eventually for an emergency use authorization. So, when we got this news, we all did the happy dance. I think this is really good news for all of the vaccines moving forward. This is proof of concept. It looks highly effective. And so if you have a vaccine that produces good neutralizing antibody, there are reasons to be optimistic that it will actually prevent infection. So now that, we're well on the way, it also raises a tremendous number of questions. And so what happens to participants in other trials? I'm currently enrolling for AstraZeneca, and everybody wants to know, well, if Pfizer is licensed, can I go get it? It's a good question. Who gets the vaccine first when it gets to be licensed? You're not going to have enough for everybody all at once. We also wonder about their antibody will be durable, and will we need revaccinations at some point, and at what interval? And would you use the same vaccine, or is it okay to use different kinds of vaccines? So there are a tremendous number of questions that will need to be worked out. It is an active topic with the NIH to try to figure out how to get more information because we clearly need more than one vaccine. If you think about the Pfizer vaccine, it is needs to be stored at -80, so maybe that's not the best vaccine for the developing world where that could be a real issue. So I've raised some questions. I don't have the answers to all of them, and I'll just stop there and see if anyone else has any questions.
That's great.
Thanks, Anne, for that. Let me just start out with a couple of questions. In your experience with vaccines, there have been other vaccines like the Averon vaccine, which presumably because they had a cold chain issue, it was harder to roll out. Between the problems of having two vaccines, some of the reactogenicity, and the difficulty of distributing the Pfizer vaccine with the cold chain problem, do you think this is realistic that this is going to have an impact in the next 12 months?
I do. I think that there are a lot of smart people that can help to solve the cold chain issue. And again, it just may not be the best vaccine for every segment of the world, because I do think that the developing world has some challenges. But in many developed countries, having a -80 freezer is solvable. And, how to then make it so that it could be at health clinics and at your local pharmacy. I feel like that problem should be solvable. The science of making the vaccine, is one hurdle, and this was tremendously good news that it is solvable. It's also possible it may not actually need to be stored at -80. This is where they start. I don't know if they've done all the studies to really see if, it can tolerate some lower doses. Lower temperatures.
Maybe the pharmacies can get those smaller if they make them -70 refrigerators, much like kids had in their dorm rooms to keep beer cold or something like that.
Mr. Frosty.
Yeah. I had a question about the Pfizer study real quick. So you covered the data nicely, but what I can't get my head around is that the Data Safety Monitoring Board met. They determined that there was a 90% efficacy. They announced it, but they didn't stop the trial. I assume that's because they need 2 months of safety, but why announce it now when you can't take the placebo group and put them on treatment? That doesn't seem standard for what we normally do.
Yeah, I can't speak to Pfizer's decision to announce it. That wasn't the Data Safety Monitoring Board's decision. What the company does to announce it. I see there's good aspects and bad aspects to that. And, I think that it's kind of the shot in the arm, ha-ha, that sometimes we need to keep us going. I mean, these are dark times, and, many people have been working, every second of every day, not knowing that we'll be successful. And, we needed this good news. I also hope that it might be used to take those people that are having pandemic fatigue and kind of like, well, why even bother wear my mask or socially distance? It's hopeless. They're never going to have a vaccine. Well, we will have a vaccine. And so they should be motivated to continue to be safe until we get to that point we can distribute it. But the reason that they announced it, maybe they needed a boost in their stock prices. I don't know. I actually like Pfizer. I've worked with Pfizer for many years.
I didn't. Yeah, I just never seen that.
I don't know what they're thinking.
And can I just ask you one question about ending this? Do you know whether the protocol is written so that the placebo recipients immediately get the vaccine if, in fact, they show efficacy? Because if that's the case, you would think this is the time to give it to them.
It is written in a somewhat not ambiguous, but it says that it will be offered to the placebos at the point with discussions with the FDA and the regulatory bodies. So they wrote it in a way where they needed to say that they're going to discuss it with the FDA. That the Data Safety Monitoring Board recommended they keep going with the study. So really, per protocol, they need to keep going with the study. And again, I think you can make a good argument about safety. That, yes, it looks incredibly good. That, 90% of the cases occurred in the placebos versus the vaccinees. And I think everyone is aware of the pressing need for an effective vaccine and don't dilly-dally. But, before you go from 45,000 to millions of people, I think they just want to be a little bit more measured and get that extra safety data.
All right, so let's talk about that. So by my back of the envelope, I'm guessing around 9 of the 94 folks in the vaccine group, 9 of the 94 failures or infections occurred in the vaccine group. So the key question is going to be, what are the correlates of immunity that maybe weren't present in them? Because as I understand it, all of them, the clock didn't start ticking for the actual outcome until after the second dose was given and the 7-day period after. So there were 94 cases, right? So assuming that those correlates we can figure out later, to me it seems like there's better news here than might meet the eye, in the sense that you showed nicely how almost all these vaccines have similar antibody production against spike protein, et cetera. So it tells me that it's very possible that to varying degrees, many of these vaccines will be efficacious, which means that we might be able to use something where cold chain is impossible, and we might have opportunities to really, if not, for lack of a better word, flood the market with more vaccines, assuming safety is relatively equal. Do you share my optimism, or I just COVID blind right now?
You've been drinking the Kool-Aid. No, I share your optimism. I think this was very good news that antibody is something we can generally measure. We can look for protective levels. We can also look that, maybe those 9 failures were very soon after the second vaccination.
Yeah, I was wondering that.
So that, they didn't present a lot of the granular data that I think we'd all like to know. But hopefully that's coming, and that, I think they're going to get to 164 cases pretty quickly.
Should be this week.
Well, yeah. So, they're thinking, it's sort of like December is going to be where they really analyze the data. And so it's not that much time we need to wait and look at but yes, I think that sometimes, not everybody hopefully going forward is going to have to do a huge efficacy trial. Maybe they can show safety data and they can show that it meets a correlate of protection.
I think Swindoll had a question.
Yes, thank you. That was a terrific talk. I just had a question maybe relevant for this audience about inclusion of people with HIV in these vaccine studies. So on clinicaltrials.gov, it says in the Pfizer study, they were excluded from phases 1 and 2, which suggests maybe in phase 3 they were in there, and maybe ultimately we'll learn something about how the vaccine performs in people with HIV.
Yes, and this again is a topic that's been tried to be harmonized throughout the big phase 3 studies, although Pfizer didn't participate, that people with stable HIV disease and controlled viral suppression are included. So in the Astrazeneca study, if you have adequate viral suppression and you're stable with a CD4 count over 200, you can be in the trial. It is my recollection that that was true for the phase 3 Pfizer trial as well.
So there are two other questions that are similar coming in from the audience, and they're kind of related, and I think we can quickly go through that. But an issue about what does a 90% mean, and when you don't know about the exposure, but what this really is just a proportion of those who got vaccine getting infected versus those who got placebo, and that's the 90%, right?
Right. You don't really need to know exposure. It's event, right. Placebo versus vaccinees, and you just count up the number of infections.
Yeah. So what about young people? And let's say these come out, and you mentioned in your last list of questions who first and all that, and I've seen arguments of saying, well, treat the very young because they're the ones who are going to be spreading it more versus the most vulnerable. So do you have a position on that?
Well, I think that they're developing plans that sound sensible to me to start, that healthcare workers are going to be first in line for rollout of a vaccine. And then following that would be frail older people, people with underlying conditions, because we know that they get sick and are the most likely to be hospitalized and die. But the point you bring up is an interesting one, and that's the other thing we don't know about these vaccines, of whether if they produce sterilizing immunity. So I told you that it's 90% effective to prevent symptomatic PCR positive disease, but we don't actually know if it's going to decrease shedding. I mean, we think it will, but whether or not there will be, it doesn't prevent asymptomatic individuals. And so we do have a lot to learn. And I think that it only makes sense eventually if it decreases shedding and illness, that you would start to vaccinate young people. I know there's interest in the vaccine trials are moving down in age to teenagers and then to older children. And I have to say we've had a little outbreak in our own unit, and somebody who is 24 and in good health is pretty darn symptomatic. So not everybody just has a sniffle. So I think it's worth considering all the age groups, but you have to prioritize certain people first.
And you might also mention there are, as you implied, other effective vaccines that do not prevent asymptomatic carriage and transmission. Do you want to say just something more specific about which those are, like meningococcus, I presume?
Well, and there was a concern with the pertussis vaccine that it was preventing symptoms, but not giving sterilizing immunity, and therefore it was perpetuating sort of a new blip in the epidemic. So it really is a real concern, but this virus is so darn infectious. It's just spreading all the time everywhere. And so if you only have a vaccine that prevents symptomatic disease or severe disease in older people and groups at risk, then that's still a good thing. It's not ideal. You'd love to have sterilizing immunity to really decrease transmission, because obviously that's important to keeping it away from frail and vulnerable people. But, one step at a time, I think.
All right, well, I guess, go ahead, Mike. Go ahead. I was just going to say, at this point, I think we've sort of come to the hour. So if you have questions, you can put them in the Q&A. But again, Ann, thanks very much for a great talk. And Dr. Sag has said he's going to give us our concluding remarks.